All right. Good morning, zoology 2700 enthusiasts. Welcome to your second Friday and the end of the little things. This is where we're going to say adieu to the protozoan diversity. We have a couple of more groups to go through today. This is going to be more like Monday's lecture, where we go through a survey of some of the diversity, the protozoan diversity that we share the planet with, and some of the apomorphies that define these groups, these hopefully monophyletic groups. So let's start out by saying, put your hand up in the air, pat yourself on the back. That was really good filling in of the word finding here, such that I wish to shave it, as a Sean Connery would say, if he were still alive. All right, so there's a couple of other things that I want to take care of before we get going here. And the first is to remind you that if you haven't seen it lately on CourseLink, there was a note put there on the announcements on the homepage by Sherry yesterday to, if you were not able to purchase your activity kits. There's only a couple left, but if you did not purchase one and you want to, could you email Sherry ASAP so that we can put some others together to make sure that you get them. And the other question that I had was just about, you can use your annotate tool there. Tell me about what you're going to do this weekend. This is a Friday. I think it's important for your mental health to remember that it's a Friday, because the difference between Fridays and Mondays and Wednesdays is kind of being homogenized by the in pandemia, the land of pandemic. So I like this. Horse riding, that's awesome. Celebrating your dad's birthdays. Try to understand MCB. Oh, good luck. Make sure that you do something. If you're trying to see, if you have to spend some time understanding a course, please make sure that you take some time for yourself. Do something physical. Do something enjoyable. Do something that gives you a return and makes you happy. Right, Dr. Jacobs?
Can I read what the plans are?
Yep.
I'm sorry, I'm interrupting. It's
Yep.
going to be a frequent thing.
This is like a guest lecture. All right, take care.
Oh my goodness, Bernie!
Yeah.
Hi. Mwah. I hope you're all well.
Dungeons and Dragons, absolutely nothing. Snowshoeing. We are going to take advantage of the fact, yes, it's a birthday, and Dad's birthday.
Oh.
That's awesome.
Okay.
Hooray to everybody's birthday in January. So, what are we going to do? We're going to be with the kids and trying to write an exam or a quiz for you guys. That's what I'll be doing. And then also taking advantage of the rink that we have in the backyard. And we are going to be kind of moving our kids along in how they learn to skate, which has been a wonderful part of having everybody at home. So I'm going to save that as well. And then I'm going to say thank you for all of those annotations. And with no rudeness involved, I'm going to clear them. And we're going to keep going. But we're not done with Bernie yet, because I wanted to remind you that we're going to be talking about, as I said, we're going to be leaving the protozoans today. But we're going to be rejoining the Metazoans. And so there's a couple of chapters. This is going to bridge. So you've been spending time in chapter 3 with your textbook, reading through kind of the key points, doing a graze, not like trying to gorge yourself with all of that information, but touching on the points. Salut, Jack. Ça va? Touching on the high points that we mentioned in class together here. And so to that grazing menu, I'm going to start digging up chapter 4 and 5. And that's actually not going to be true only. Chapter 4 and 5 talk about a lot of neat things. I mentioned them there, animal architecture and body plans. That's Dougie the dog that you can hear maybe barking in the background. And an introduction to animals. And in both of those chapters, we're going to talk about things that will come up again and again and again. Some of them will be specific morphological things that in Brusca are contained in one chapter. And some of them are bigger conceptual things like surface area to volume ratios and how that's important.
I colored something and you could see it when you're done.
Ok, tu peux me montrer peut-être, mais je vais continuer de parler avec mes amis ici, ok?
Okay, it's just
Ok, c'est bien. So, chapter 3, 4, and 5 are now on the table. Hooray. So this is where we've been spending the time. We took a lot of time with the apicomplexans on Wednesday. That was like a deep dive that we do. This will be more in the late night pantheon, their pantheon of late night TV shows. The other day was kind of a John Oliver episode where we took one thing and dove deeply into it and talked about a whole bunch of different aspects of it, from society to morphology. Today is going to be more of a grazing, so it's more of your Seth Meyers, Stephen Colbert, where we're going to hit on a bunch of things all throughout today's lecture. And in neither case is it meant to be that they are funny, because moi. So in the pre-lecture for Lecture 5, I talked a bunch about the ciliofferens. Remember Latin for eyelash, the cilia with the beating metachronal waves moving them across the landscape. And there's a couple of aspects of the cilia that I didn't mention in that pre-lecture that I wanted to take some time, as we share some time today, to mention, because they're kind of amazing. And one of the principal aspects that I wanted to share was to talk about how these tiny, tiny protozoans, without any particularly hard cellular structure, can defend themselves. So here's a couple of terms here that you might see appearing in a future crossword or a word find, but these are different defense mechanisms. So there are extrasomes that they can use. So these are membrane-bound defensive structures. There are trichocysts, which you're going to see in a second, which are nail-like spikes that are presumably used in defense. Toxocysts that can be used to extrude threads that can be used for defense and or capturing prey. Mucocysts, which do what you might imagine and extrude mucus filaments, again in defense or prey capture. And then haptocysts, which are harpoon-like structures. So a whole range of different adaptations that ciliofferens have for both defense and potentially also for prey capture. So this is a view. They're going to get two views here of Paramecium. Remember Gauss studying Paramecium to look at competitive exclusion. These are Paramecium Trichocysts that we're going to watch once in real time, and then I slow the video down so you can see it at many more frames per second. So you can see here the kind of oval shape of the edge of the Paramecium under this slide, and you're going to see the cilia beating. We're going to watch it together. So beating cilia, beating cilia, beating cilia. Oh, I've got a problem. Did you see those needles come out at the edge? Take a look here, and they're appearing now in slow motion. These are trichocysts being extruded by the membranous wall of that Paramecium. So it's essentially turning itself into it's like a porcupine defense, if you want to use a mammalian kind of analogy. That clearly breaks down, but these are spiky things that are going to then encourage a predator not to consume it. There are other things that these tiny little ciliofferens like Paramecium can do. One of the cool ones are starting to have to think about water regulation and surface area to volume ratios, and the fact that the cytoplasm, this kind of interior gelatinous bag, the cytosol that keeps these things, gives them their small shape. In aquatic environments, you could have too much water flowing inside of the cell, and so this is a small bit. These water expulsion vesicles or contractile vacuoles, you can see them in that video. They were, let's jump back to the front. Yeah, see that winking eye on the bottom at about 8 o'clock? That is a contraction vacuum. So that's pumping excess water out of the cell. And there's a lovely diagram that you can see how they look kind of exactly as you see in that video on the bottom. So these are different adaptations to living in an aquatic environment and pumping excess water out of the cell. Ciliofferens, like the rest of us, have to think about reproduction as well, and there are a couple of different ways that they go about this. One of the first things to look at here is that on the left, you see these kind of kidney, or like a peanut or a Brazil nut, and a small little poppy seed. This is a characteristic of the ciliofferens where they have a macro and a micronucleus. And so there's like a master copy, and then in the micronucleus, and then the working copies, millions and millions of copies of certain genes. Now, to reproduce that, they go through two different kinds of reproduction. They can have asexual fission, where there's transverse planes that across rows of netids. Now, you remember netids, right? This is netids are so those long planes of the basal body containing a beating cilia knit together by a basal fiber that coordinates the beating of those metachronal waves. So if you're going to split, you wouldn't want to do it across at any different kind of strange angle to those netids. You want to do it in line. So that's asexual fission. You'll see some video of that in a second. And then there can be sexual conjugation. So conjugation involving meiosis and the exchange of micronuclei. The macronuclei degenerate, but let's take a look at prior to conjugation. Let's take a look at those. So on the left-hand side, you can see fission. So this is that splitting across the rows of netids, and then asexually kind of one turning into two. Then there's video of conjugation here on the right-hand side. We're not going to watch all of that video because, as Rupert, Fox, and Barnes, remember these are the ones, the older texts that tend to wax lyrical and poetic about the diversity of invertebrate life that we share the planet with. These are the ones that talked about the metachronal waves being as wind across a wheat field. Conjugants may be blissfully fused for several hours. Bliss as experienced by a protozoan? Not sure if there's any kind of room for that, but we can anthropomorphize it, and if by anthropomorphizing we build empathy, then I would say, sure, let the anthropomorphizing in the room. And then the fission has then already completed. The conjugants will remain blissfully fused for several more hours. So, Bernie and I have a question, or what I take the vérifier le chat right now. Let's see what I'm missing. Freshwater. The FW is freshwater. Lots of joining of the chat. Hello, Dr. Jacobs. Bernie is great. I think I caught most of those questions. So onward, we continue. Please remember, drop those. I'll try and get back to the chat pretty regularly. So if there are questions as we go, make sure to grab them there. And it's also, this is preserved on course links. So those questions as they come up, if I don't catch them right away, I go through afterwards and try and read them and then give some thoughts about them later on. So this is maybe a studying resource that you can come back to as you think about your quizzes later on. And we continue. It's also a good place to tell me that my mic isn't working, or I'm sharing the wrong screen, and why are you looking at your banking information? All right. Let's take a look at, yes, that's a very studious shot. And I'm going to thank you for those, clear them all, and we continue. So you've seen this. Remember the polytomy? This is where all of the diversity that we've seen, we were just inside of the ciliates there on the left-hand side within the alveolates. And right now we're going to jump across to another group, one of the kind of 2 or 3 last of the major groups. So this is actually a sister group, if you want, the closest related, the most closely related protozoans to us are these amoeboids. Now, an amoeboid lifestyle, so there is a true amoebozoan group, but an amoeboid lifestyle, as you might have been taught, say, in the ancient past, in the 20th century when I took this course, it wasn't really a monophyletic group, because pseudopods, which are one way that amoeboids have to move, and there's different kinds of them, and we'll talk about the pseudopods and how they move with this extension of the cellular wall. That's likely an ancestral trait of a whole bunch of eukaryotic cells, and so we see it in a bunch of different lineages. We'll come back to that point in a second. So there are several different groups of amoeboid protozoans. There are the true amoebas, there's foraminiferans and actinopods, or actinopods that should have a quote around it. You'll see that later on, but the rhizarians, or the heliozoans. So, what were we talking about in terms of the amoeboid protozoans? Well, here's this kind of amoeboid creeping, and what you're looking at is an amoeba that's phagocytosing, that is eating, it's consuming a food particle. So this transition of the cytoplasm, that kind of interstructural, kind of intracellular structure between a stiff, clear ectoplasm that's called the gel, or the fluid internal endoplasm that can kind of stream out and fill one of those pseudopods, or lopopods, or phylopods. These are different words. Pod, of course, meaning arm, and what those different words are just different ways to describe basically how thin or thick the extension of the body wall is. Let's take a look at that here on this pigeon. So we've got streaming as the transition of cytoplasm, pseudopods, which we saw, these phyllopods. So we've got a lopopod there shown on the right-hand side and a phylopod, a thinner extension. Now, how do they become thinner or thicker? Often it's through protruding through a test or an external skeleton. Now, those skeletons can be proteinaceous or silicaceous, but they're basically, they become longer nets or kind of branches for sitting on top of something that you want to feed, like in particular, if this amoeboid was feeding on coffee, that would be a pseudopod reaching out and grabbing that. And if I wanted a thinner net spread across to filter many tiny cups of coffee from this room, then a phylopod or something that branches out and essentially increases my surface area dramatically to increase the likelihood that I run into tiny cups of coffee. Would that it were so simple. So there, you can see these feeding events where the amoeboid kind of streams, creates this food cup, and then creates phagocytoses it and creates a food vacuole inside as it pinches off around there. And this is from your text, figure 3.10. So a quick extension into amoeboid feeding. And now, so the true amoeboids over here in brown beside us is the opsiconts, or the animals, metazoan animals. We're going to jump into another group, and you can see how distantly related. So this is a very similar looking group that has amoeboid type feeding, but is very clearly not a monophyletic group, because for two reasons. One of them is that we have a nice polytomy at the base of this tree, and the other is that it excludes rather important groups like all of the algae, all of the land plants, and all of the mammals. If we were to make a group called amoeboids, or the amoebozoans, they would then be this kind of a group that would exclude lots of descendant groups. So, you know some of these rhizarians. So we're talking about foraminiferae and radiozoans, Foraminifera you know about, whether you realize it or not, because they have a diverse fossil fauna, and you've seen or probably seen movies that have. So if you watched Christopher Nolan, Dunkirk, there was lots of views of the White Cliffs of Dover as different planes and boats kind of took off and either left England heading towards France or were coming back from France to England in the kind of the mixed up way that he tells his stories. You saw the White Cliffs of Dover several times in that film. Those are all, now it didn't get the same kind of heading at the credits at the end of that amazing film, but there are tens of millions of foraminiferan shells of tests, fossilized tests, preserved in those White Cliffs of Dover. So they have another kind of podia called reticulopodia, so that's even a finer network of this amoeboid body that's out extended into the marine water to help them feed. A diverse fossil fauna, a calcareous test, that's the color that you're seeing there. And about 45, so they've been around for millions and millions of years, quite a successful group, and mostly marine, and you'll see them all over the world. And there's a little screen grab at the bottom there of the box that talks about the foraminifera inside of Chapter 3 of Brusca, where you can consult later on for some of the particular apomorphies that define this group. Regularians are, again, mostly planktonic. They have an organic test, proteinaceous or silicaceous. They have algal symbionts, so you'll see some bits of color in green. Now, this is a group, the Regularians, that used to be called the Actinopoda, in that it was a group that contained Regularians and Heliosomes. We don't use that word, and so I put in Actinopodia. Anytime you see quotes like that, that's essentially me bringing up a word that you might run into if you continue with your interest in these groups, but you likely won't find in Brusca. If you have the digital copy, you can go and you can actually use Control F in that text, and Actinopoda doesn't appear at all. This is one particular case which required the updating of that text through time, because our terms change and our understanding of who's related to whom changes. So they have axiopods. So we've got pseudopods, phylopods, reticulopods, axiopods. So these are needle-like, so extending even farther. that are used for prey capture, that help them float or maintain themselves in the water column. They can be used to attach themselves to different things, to different objects. They can be benthic or planktonic. And the test seems to be always organic. The Heliosomes are slightly different. You can see again with these, let's go back, with the Axiopods, but you can see silicaceous spicules extending, Axiopods extending way out like some kind of very, like a furious hedgehog. And they also have algal symbionts. So as I said, in a grazing way, in diving in deeper into some of these groups across the tree of protozoan life, this divergent tree, we've now been 3, 6, 9 different groups, and we haven't got to the animals yet. But you can see here, remember some of these, some of the relationships we do understand. So way back when we talked about the euglenozoans, the euglenids and the cnidoplasts, they're pretty closely related. The aviolites over there on the left-hand side of that tree, the dinoflagellates, the ciliates, and the apicomplexans, we're pretty confident about that close relationship. The rhizarians, they are foraminiferae and the radiozoans. Those are a well-supported group, and one that we've blown up in the somewhat recent past is the an amoeboid group that included also the amoebozoans with other amoeba-looking but distantly related rhizarians. So I've got another view of this. This is us transitioning out of the protozoans. Patrick Keeling, who's a biologist at the University of British Columbia, he had this tree published, let's say, like a long time ago, 2005, in the Trends in Ecology and Evolution, talking about large chunks of life. So if you didn't like those Trivial Pursuit pies across that flattened tree from Nebraska, here's a view at the uniconts, the rhizarians, the excavates, green algae, which we ignore, and the chromoaviolates. And you can see there in red, I've highlighted the groups that we've dipped into so far, and then in red, animals, where we're going to go for the rest of the course. That's a lot. This is a lot of material. So what I would suggest, again, we are grazing. One of the learning outcomes from this course is for you to develop an appreciation for the strategies and the diversity of the life that you share the planet with. The majority of that life is invertebrates. So we don't have the time, you don't have the time, none of us have the time to dive deeply down and to break up all of these groups. And the perspective that I often take is that if you don't take any other invertebrate courses for the rest of your career or the rest of your life, this is maybe the only chance where somebody's going to have to spend time with you, sharing their enthusiasm and sharing their wonder for some of the diversity of life and the diversity of strategies that life has attained through evolution, through time. And so as you're studying, please do not feel that you need to dig down into the rhizarians and read all of that there is in the text. If you find a group, a taxon, that I've spent very little time with, you could only positively expect that in a test or a quiz, there won't be very much about it there either, okay? If we spend more time with one group, you can reasonably presume that that's where we'll spend more time in a quiz and the kinds of questions that we'll talk about later on in the quizzes. Fair enough? Onward. So there's kind of a cheat, not a cheat sheet, but just a summary sheet of the different kind of principal 4 groups of the protozoans that we spent time with in bold, at the bottom, some of the defining characteristics that we spent some time talking about, and then up on the top, some of the different taxa that we talked about by name through time there. So, Caramel and Bernie and I will jump back over and take a look at, yes, the triangles, Okay. I'll try and do this in time. Calcareous test. When I say calcareous test, what does that mean? So calcareous derived from calcium, or calcium carbonate in water, and calcareous, so it's a test, and the test means like an exoskeleton. So it's essentially a hardened surface that it's deriving from the water, based on chemicals in the water. Do puffins feed off of these? Is that why there are so many puffins on the cliffs of Dover? Yeah, puffins feed on a whole bunch of things. Probably not specifically on these, but on the things that the other, for instance, crustaceans. that are eating these things. So they're definitely dependent upon them, but they're 1 or 2 steps removed in a food web from a food web perspective. Do the colors of that picture have a specific meaning, like similar characteristics or something? I think that, Durza, yeah, you're referring to the tree. So the red triangles are me just calling attention to where we have spent time. And the colors on the tree refer, let me jump backwards here. And yes, so the colors there on the tree, if we look at, I love the fact that the mustache from the cat probably becomes cilia there on the ciliates, with a floating cowboy hat above it. Those colors refer to monophyletic groups. So the brown, the uniconts, the blue, the fornicates and the preoxalates. The fornicates, we spent very little time in that blue end. The kind of khaki clade at the left-hand side, we spent a lot of time there with the dinoflagellates and the apicomplexans and the ciliofferans. A lot of time there, didn't we? A lot of time. Okay. Let me jump back to the cat and keep checking here. Water excretion vesicule. So that's that kind of blinking eye in the video. So it's a osmoregulation. It's a tool for osmoregulation. All right. Let's. I thank you for your drawings and the kinging. Save that. Clear all the drawings. And onward. Metazoans. So for the next 10 or 15, 20 minutes, what we're going to be talking about are animals, and we're going to talk not about a particular taxon today. I'm going to try and hold myself back from starting a taxon review for the first groups that we talk about in this Dunn phylogeny, but I wanted to bring up again to your attention the Dunn phylogeny. Everybody say. So this is the complex version from Dunn and Friends, published in the Annual Review of Ecology and Systematics a few years ago. This is a lot of information here, but it's actually really simple, and that's why I present it, and I will continue to present it as we bounce through this tree for the rest of the term, up until the spring. So we've got nodes that are strongly supported there in yellow. Right? We have different terms kind of turned at right angles, so you'd have to turn your head like this to read them. The bilaterian, the animals, those nodes, and those are the names associated with those nodes. We'll see a simplification of just those nodes, well-supported nodes, in a second. But here with the 32 tips, the diversity and the names extended across them, these are the groups, the taxa, that we're going to jump in and out of for the rest of the term together. We then got the number of species, an estimated, inevitably an underestimate of the currently described diversity. You can see most explicitly down at the bottom with the ants. That's shown with the arthropods when it's greater than 1.2 million species. And you can see the range of diversity there within these phyla extends from the placozoans here, which we'll talk about probably next week, I believe, with one named species, a phylum of one, the loneliest phylum, to the arthropods, the least lonely phylum, more than one, and this is insects, so you can, and crustaceans, and many, many, many fascinating things. And then you've got the some silhouettes on the right-hand side, and then your color stamps about different strategies associated, ways of life that are seen within each one of these phyla. So you can see when you look at the arthropods down at the bottom, they are found in marine environments, freshwater environments, terrestrial environments, and they live parasitically. Those are the colors from left to right, from blue to green to orange to red. And then you can see the range of kind of strategies across all of animal life. Now, one of the biases that I'd like to call your attention to, that I have, and that inevitably you have, is that the taxa in orange, the terrestrial life, that tends to be the things, those tend to be the taxa that you know about, that probably you can draw, or that you have photographs of on your phone, or that your history on Netflix shows you thinking about. The ones that are marine, less so. And the ones that are not, and I'm going to skip to the simplified view here in a second, that are not bilaterian, or that are not deuterostome, those are the ones that we're increasingly, our bias increases, our bias against them increases. We understand the world as a bilaterian thing. We are a deuterostome, so we're in this craniotic clade here. That's our home. You can see the neighborhood around us has some interesting things. It includes those starfish that are in your freezer. But then look how diverse the city of animal life is around us. Things that live outside of our neighborhood, but in the same unicont city of life. And radial or absent symmetry being absent, these are groups that we know very little about. There are some unifying factors, though, across this diverse group, the bilaterians, the deuterostomes, the protostomes containing ecdysozoans and spiralians. Now, some of the things that tend to unify this group, we'll talk about here in a second, that they tend to be mobile, tend to be heterotrophs, so we're not dealing with phototrophs or autotrophs any longer. We tend to be multicellular, that we develop from embryos, that there is differentiation of our somatic cells, and that differentiation inevitably leads to specialization. We have tissues that can be special, where specialized cells are bound together to perform specialized tasks. The epithelium is one of that we'll talk about. A really critical evolutionary advantage or occurrence that turns the world essentially into inside and outside. And that's a critical stage if we're going to start specializing. We have connective tissue and collagen, perhaps the most structural protein, the most important structural protein for animals. And because whenever there's a chance to hear Attenborough talk, we take it. Here he is, talking for a minute or so about collagen from the BBC documentary First Life.
At a microscopic level, small cells.
So I can't cheat and look at. So there's a link there at the bottom, and I'll put this up in the chat later on. When the video's starting and stopping, that's me having to have my slow brain remember the fact that once I'm showing a video here, as soon as I try and say answer a question in the chat at the same time as the video, it stops the video. So Smith will try very hard to remember that. Super important. And one of the things that Attenborough is talking about there in this video, the full episode of which you can find at that link below, is the fact that associated with increasing complexity in the evolution of life on Earth is the increase in concentrations of oxygen, and the fact that allowed life to become both multicellular and construct lots of different things that collagen helps us construct by being essentially the critical building blocks for those things. Since I'm here looking at the chat, the orthonectidia, there's a small number. I don't know why that number isn't there. It's in your text actually, and or in your lab book. Would you say that the Dunn phylogeny is more important to know, or the ones that have been shown with the pie slices? More is a strain. I don't know if I would use the word more, but we're gonna. So we've had kind of 3 lectures where I've shown that 3.53 from Brusca, the phylogeny that includes all the different protozoan groups. We're now going to have 30 more lectures referring to the Dunn phylogeny. So just in time that we have to emphasize them, the Dunn phylogeny will be referred to more often. But I think it's important to remember where that Dunn phylogeny sits within the larger phylogeny. And if you're worried or concerned about memorizing one of them, don't, because I would expect you to draw only at most the simplified version. And if I wanted you to refer to the simplified version of the Dunn tree, that second one, the tattoo that is in your lab kit. And if I wanted to make reference to the alveolate clades, I would show you that figure in the question. Extracellular test, so the test outside of the body, so as a calcium carbonate hard shell on the outside of the cellular membrane. That's extracellular test. Cool. And oh boy, here we go. So, multicellular life. We stopped for a second and took some questions and had a chance to chat about why, why were protozoans small. We're going to take maybe think of those threads that you thought about with your groups in the breakout rooms in Zoom earlier this week, and maybe pull some of those threads together, and you're going to start sewing or weaving a new sweater here, because what I'd like to do is talk about advantages of multicellularity. So I'm going to put you in breakout rooms for about 5, maybe 4 or 5 minutes, and we're going to talk about, I'd like you to talk about what those advantages are that you come up with. Why has multicellular life been so successful? What are the advantages? Spend 2, and then go to the same link for the Google spreadsheet and tell me about some of the results of that discussion with your group. So here we go. Open up all the rooms. Hi, Sherry.
Good morning. How are you? It's all good?
Yeah, I'm told it's Friday.
I know. Thank goodness.
So what are you up to this weekend?
I'm trying to figure out what the students are going to do next week in invert.
Me too. Me too.
Yeah.
The analogy that keeps coming up in my brain is a surfing one, where it's like the wave is breaking. We're kind of on the face right now. We had 1 week down, 2 weeks down, and then you look behind and the wave is starting to crash, and you're like, ah.
Yeah, it sort of threw a wrench in the plans when we didn't get those slide scans. So
Yeah,
I'll take some pictures and do it that way. But
okay. Let me know if you want the GigaPen thing would be pretty easy to do if there were particular regions or anyhow. We talked about that before, but
yeah, and I think maybe later on when we get to some of the bigger stuff, especially, it would be very cool.
okay.
I know Carolyn had talked about maybe trying out some of the Z stacking images and stuff for some of our thicker slides once we get into the crustaceans and stuff like that.
Cool.
So, it's just a time crunch this week more than anything. So it's, and we're looking at the amoebas and things like that. So it's, it's just trying to figure out how to keep them engaged.
Yeah.
So, but I did want to mention, and I was going to send you an email, but I've got you here now, is I had a meeting with Jenny yesterday because she's the TA in charge next week.
Yep.
And so she's going to do a thing about the face mites and showing the students how to use bold. And so she thought she would send them into a breakout room and get them to find just something that interests them and see how many records there are, just to get them sort of talking to each other and using the system. Just a quick activity, that's all.
Okay, that's great. That's going to happen later on as well, but
yeah,
super.
That's. I knew that you were going to do it later on, but I thought, you know what, it might be good to just introduce them to it now.
Yeah. I think it's totally a good idea.
Yeah. Okay.
Hooray.
Yay.
Hooray. All right. Maybe I should go tell them 1 minute till we come back.
Awesome. Take care.
All right. Oh, look at all those answers. That's awesome. All right. Hello, everybody coming back. I hope the breakout rooms were clean. A nice ambiance. come on back together and let's take a look at all of the great, I was kind of seeing from the outside lots of the great ideas that you've got here. Okay. All right. Welcome back, everybody from the breakout rooms. And I've shown, and you should be seeing, please tell me if you're not, the results of your Google Sheet there. So what are some of the advantages that you came up with as experienced biologists for being multicellular? So let's make that a bit bigger for the old person amongst us. If the cell dies, the entire organism does not die. I like that. Starts with the metaphysical. Cellular support. They can work together to fight off a virus or injury. Not limited in size by the limitations of being a single cell. Yes, so diffusion and how far that can get amazing. Complexity is permitted. Can grow larger and reach larger sizes. More complex functions. Increased number, and because of that, niches that are accessible with specialization. I love that one. Can live longer, yes. Differentiation, so specialization again, less of a limit on body size. Cell specialization, more adaptable. Differentiation. Oh, you're not seeing the document, are you? Boo. How about this? Well, you can actually, if you're in the document, you're seeing, I was reading through, and in the PowerPoint that you get later on, I will put that Google Doc, the results of that Google Doc. But pat yourself on the head. You've come up with by far the majority of them. And in the interest of kind of keeping going until and letting you go at the time that we're supposed to let you go today, many of those differences that you came up with are correct. And many of them, and I have my own list that you'll see in a second, many of them are due to differences and changes in the surface area to volume ratio. And that surface is critical because it's the interface between the organism and the cell. And volume, of course, is different than surface area, one increasing to the square, one increasing to the cube. The number of reactions that can take place in the cell is a feature or a function of the volume. But to contribute to those reactions, any materials, food, have to enter cell across a membrane, so a limitation of the surface area. So the surface area controls how much can enter the cell. So a larger cell has a smaller surface area to volume, and at some point the surface area is too small to allow sufficient materials to enter the cell. You know this, right? You've talked about this back into high school, so you know about these different cubular organisms where we've got length increasing to the square and volume increasing to the cube. And you've probably thought about how that affects warm-blooded animals. And if you haven't, I'm going to show you a short video here in a second about largely how to make an elephant explode with science.
Imagine a fleshy cube.
And tell me if you can't hear this, but you should be able to.
If you double the length of its sides, its surface and volume do not double. In fact, the surface is now 4 times the original size, and the volume of the cube 8 times the original size, which is called the square cube law, and has been a law in nature for billions of years. So why is this a problem for big animals? Because heat can only leave an object via its surface. So if we make our mouse the size of an elephant, or 60 times longer, it has 3,600 times more surface from which to lose heat. But it has 216,000 times more volume, filled with trillions and trillions of new hot mitochondria that produce more heat. A lot more in size, not that much more skin. Our mouse is very dead, very fast. To go the exact opposite way. If you're small, you have a lot of surface area compared to not a lot of volume. You don't have a lot of cell ovens and lose the heat they produce very fast. So very tiny mammals came up with a very extreme solution.
What's your extreme
Meet
solution?
the Etruscan shrew, the smallest mammal on Earth. A mole-like thing that's more closely related to hedgehogs than to mice. With a body length of 4 centimeters, it only weighs about 1.8 grams, as much as a paperclip. It's a tiny, ridiculous being. It would basically cool off immediately, so its cells run on overdrive to stay warm. Its tiny ovens are filled at maximum capacity. Its heart beats up to 1200 times a minute, and it breathes up to 800 times a minute. This creates an extreme need for energy, so the shrew has to eat constantly.
That's you.
After only 4 hours without food, it starves to death.
That's not quite you. So, here's my exemplar, Etruscan shrew, or miniature dachshund. This is Lily. Say hello, Lily. Hello. Or Lick. Yeah. And so, thinking about these are, I'm kind of showing you that video because I love the video, and I love thinking about the limitations of large and small, or characteristics of being a large and a small, and constraints of being large or small. But as it pertains to invertebrates, I think there's other ways to think about surface area to volume, many of them, but one of the ways is as it relates to strength. Now, you've seen these kinds of things. You might not have seen a weaver ant lifting 500 times her weight as is in that image, but you've seen ants carry ridiculously large amounts of large masses. And you've seen Olympic-style humans carry comparatively small amounts. What I'd like to do is join you or send you to the breakout rooms one more time and have you, before you leave, because you've got to go in a couple of minutes, but discuss for a couple of minutes why the invertebrate is stronger than the vertebrate. Why is the ant stronger than the person? How and why? Put those into the Google Doc, and then we'll talk about how strength is related to surface area to volume in a second, or when we start on Monday. So there you go.